Good afternoon, everyone. I am Leia Abigail de la Vega. Together with my groupmates, we're here to tackle about biodiversity. So, what is biodiversity? It is the variety of life on Earth, in all its form and all its interaction. If that sounds bewilderingly broad, that's because it is. Biodiversity is the most complex feature of our planet, and it's the most vital. Without biodiversity, there is no future for humanity. The term biodiversity was coined in 1985, a contraction of biological diversity, but the huge global biodiversity loss is now becoming apparent crisis equaling or quite possibly surpassing climate change. More formally, biodiversity is comprised of several levels, starting with genes, then individual species, then communities of creatures, and finally entire ecosystem, such as forests or coral reefs, where life interplays with the physical environment. These myriad interactions have had made Earth habitable for billions of years. A more philosophical way of viewing biodiversity is this: it represents the knowledge learned by evolving species over millions of years about how to survive through the vastly varying environmental conditions Earth has experienced. Seems like that, experts warn humanity is currently burning the library of life. Just how diverse is biodiversity? It is mind-bogglingly diverse. The simplest aspect to consider is species. About 1.7 million species of animals, plants, and fungi have been recorded, but there are likely to be 8 to 9 million and possibly up to 100 million. The heartland of biodiversity is the tropics, which teems with species. In 15 hectares or 37 acres of Borneo forest, for example, there are 700 species of tree, the same number as the whole of North America. Recent work considering diversity at the genetic level has suggested that creatures thought to be a single species could in some cases actually be dozens, then add in bacteria and viruses, and the number of distinct organisms may well be in the billions. A single spoonful of soil, which ultimately provides 90% of all food, contains 10,000 to 50,000 different types of bacteria. The concern is that many species are being lost before we are even aware of them or the role they play in the circle of life. Just like this flying insect, I know that 75% of insects were lost in the last 25 years in Germany, according to a recent study by Frances Marriott. But there's a possibility that this flying insect will currently have a massive growth.
Biodiversity has three components, namely the species diversity, genetic diversity, and ecosystem diversity. Genetic diversity refers to the range of different inheritable traits within a species. In a species with a high genetics diversity, there would be many individuals with wide variety of different traits. Genetic diversity is a critical for a population to adapt to a changing environment. Example, birds. Birds comes in different variety of size, color, are controlled by genes and either increases or decreases the genetic diversity, in which in greater diversity increases the odds of survival of the species or population as ecosystem changes. Species diversity is simply the number and relative abundance of species found in a given biological organization or a place with a high species diversity. And as a species diversity increases, the most likely the health of the ecosystem also increases.
Ecosystem diversity, the number of distinct ecosystems within a defined area. It describes the assemblage and interaction of species living together and the physical environment of a given area. It relates varieties of habitats, biotic communities, ecological processes in biosphere. It also tells about the diversity within the ecosystem. For example, the landscapes like grasslands, deserts, mountains, and etc. It shows ecosystem diversity. The ecosystem diversity is due to diversity of niches, trophic levels, and ecological processes like nutrient cycling, food webs, energy flow, role of dominant species, and virus-related biotic interactions. Such type of diversity can generate more productive and stable ecosystems or communities capable of tolerating various types of stresses, for example, drought, flood, and etc.
Hello everyone, I'm Julia. But before we proceed to the next part of this lesson, I want you to observe these two images. What can you say in the first picture? How about the second one? Great! Looks like you already have an idea about the next part of our lesson for today. At this time, we were going to talk about high and low biodiversity. So what is high biodiversity? Does anyone here has an idea about what is high biodiversity? Well, high biodiversity is a habitat or ecosystem that has a high number of different species. So when biodiversity is high, it means there are many different types of organisms and species. Example are places in the tropic regions, like the waterfalls. You can see different kinds of plants and animals or insects, I think. As well as the second picture, under the sea you can see different kinds of fishes and coral reefs. So when we say high biodiversity, it has greatest number of organisms living in a certain ecosystem. Also, it provides shelter, water, and food to many species. So, it sustains life because it provides the needs of an organism. In results, to a stable ecosystem with lesser rate of species loss. So, that is for high biodiversity.
Low biodiversity. In this lesson, we will be tackling the low biodiversity, the causes of low biodiversity, and the ecological effects of low biodiversity. So, what is low biodiversity? Low biodiversity, also called loss of biodiversity, a decrease in biodiversity within a species, an ecosystem, a given geographic area, or Earth as a whole, describes a decline in the number, genetic variability, and variety of species and biological communities in a given area. This loss in variety of life can lead to a breakdown in the functioning of the ecosystems where declines has happened. The idea of biodiversity is most often associated with species richness or the count of species in an area, and thus biodiversity loss is often viewed as species loss from an ecosystem or even the entire biosphere. However, associating biodiversity loss with species loss alone overlooks other subtle phenomena that threaten long-term ecosystem health. Sudden population declines may upset social structure in some species, which may keep surviving males and females from finding mates, which may then produce further population decline. Declines in genetic diversity that accompany rapid falls in population may increase inbreeding or mating between closely related individuals, which could produce a further decline in genetic diversity. Even though a species is not eliminated from the ecosystem or from the biosphere, its niche or the role the species play in the ecosystem it inhabits diminishes as its number falls. These are the examples of biodiversity loss, the deforestation in Australia, and the bleached coral seascape. The primary drivers of biodiversity loss are influenced by the exponential growth of the human population, increased consumption as people strive for more affluent lifestyles, and reduced resources efficiency. Causes of low biodiversity. First is the natural biodiversity loss. An area's biodiversity increase and decrease with natural cycle. Seasonal changes such as the onset of spring create opportunities for feeding and breeding, increasing biodiversity as the population of many species rises. Biodiversity loss is typically associated with more permanent ecological changes in ecosystem, landscapes, and the global biosphere. Natural ecological disturbances such as wildfire, floods, and volcanoes eruptions change ecosystem drastically by eliminating local population of some species and transforming whole biological communities. Such disturbances are temporary because natural disturbances are common and ecosystems have adapted to their challenges. Next, human-driven biodiversity loss. Bio- Biodiversity losses from disturbances caused by humans tend to be more severe and longer lasting. Humans or Homo sapiens, their crops and their food, animals take up an increasing of Earth's land area. Half of the world's habitable land has been converted to agriculture, and some 77% of agricultural land is used for grazing by cattle, sheep, goats, and other livestock. This massive conversion of forests, wetlands, grasslands, and other terrestrial ecosystems has produced a 60% decline on average in the number of vertebrates worldwide since 1970. Biomass related relative biomass on Earth. The planet's biomass is classified by kingdom of life and other major groupings, and the size of each group's relative footprint is displayed using gigatons of carbon as a common measure. Researchers have identified five important drivers of biodiversity loss. First is the habitat loss and degradation, which is any thinning, fragmentation, or destruction of an existing natural habitat, reduces or eliminates the food resources and living space for most species. Species that cannot migrate are often wiped out. Next, invasive species, which are non-native species that significantly modify or disrupt the ecosystem they colonize, may outcompete native species for food and habitat, which triggers population declines in native species. Invasive species may arrive in new areas through natural migration or through human introduction. Overexploitation, which is the harvesting of game animals, fish, or other organisms beyond the capacity for surviving populations to replace their losses, results in some species being depleted to very low numbers and other being driven to extinction. Population, which is the addition of any substance or any form of energy to the environment at a rate faster than it can be dispersed, diluted, decomposed, recycled, or stored in some harmless form, contributes to biodiversity loss by creating health problems in exposed organisms. In some cases, exposure may occur in doses high enough to kill outright or create reproductive problems that threaten the species' survival. Last, climate change associated with global warming. which is the modification of Earth's climate caused by burning of fossil fuels is caused by industry and other human activities. Fossil fuel combustion produces greenhouse gases that enhance the atmospheric absorption of infrared radiation or heat energy and trap the heat, influencing temperature and precipitation patterns. Ecological, ecologists emphasize that habitat loss, typically from the conversion of forests, wetlands, grasslands, and other natural areas to urban and agricultural uses, and invasive species are the primary drivers of the biodiversity loss, but they acknowledge that climate change could be a primary driver as the 21st century progresses. Ecological effects. The rate of biodiversity loss is most pronounced in species whose populations are decreasing. The loss of genes and individuals threatens the long-term survival of a species as mates become scarce and risks from inbreeding rise when closely related survivors mate. The wholesale loss of population also increases the risk that a particular species will become
Measuring biodiversity species richness. Species diversity is determined not only by the number of species within a biological community, species richness, but also by the relative abundance of individuals in that community. Species abundance is the number of individuals per species, and relative abundance refers to the evidences of the distribution of individuals among species in a community. Two communities may be equally rich in species, but they differ in relative abundance. For example, each community may contain 5 species and 300 individuals, but in one community, all species are equally common, and 60 individuals of each species. While in the second community, one species significantly outnumbers the other 4. These components of species diversity respond differently to various environmental conditions. A region that does not have a wide variety of habitats usually is species poor. However, the few species that are able to occupy the region may be abundant because competition with other species for resources will be reduced. Trends in species richness may reveal a good deal about both past and present condition of a region. The Antarctica continent has few species because its environment is so inhospitable. However, oceanic islands are species poor because they are hard to reach, or as is the case with the lesser Sunda Islands in south central Indonesia because they are of rather recent origin and organisms have not had enough time to establish themselves. Biodiversity loss is most often associated with species richness, the count of species in an area, and thus biodiversity loss is often viewed as species loss from an ecosystem or even the entire biosphere. See also extinction. However, associating biodiversity loss with species loss alone overlooks other subtle phenomena that threaten long term, and also solution to biodiversity loss occur in tropical environments where species richness and biodiversity are much higher than in ecosystems closer to the poles. Measuring biodiversity. Looking in this picture, although examining counts of species is perhaps the most common method used to compare the biodiversity of various places. In practice, biodiversity is weighted differently for different species, the reason being that some species are deemed more valuable or more interesting than others. One way this value or interest is assessed is by examining the diversity that exists above the species level in the genera, families, orders, classes, and phyla to which species belong. For example, the count of animals species that live on land is much higher than the count of those that live in the oceans because there are the huge number of terrestrial insect species. Insects comprise many orders and families, and they constitute the largest class of arthropods, which themselves constitute the largest animal phylum. In contrast, there are fewer animal phyla in
terrestrial environments than in the ocean. Measuring of this variety, called species richness, is the count of species in an area. Colombia and Kenya, for example, each have more than 1,000 breeding species of birds, whereas the forests of Great Britain and of eastern North America are home to fewer than 200. A coral reef of northern Australia may have 500 species of fish, while the rocky shoreline of Japan may be home to only 100 species. Such number capture some of the difference between places. The tropics, for example, have more biodiversity than temperate regions, but raw species count is not the only measure of diversity. Furthermore, biodiversity encompasses the genetic variety within each species and the variety of ecosystem that species create. More generally, areas differ in the biodiversity of species found only there. Species having relatively small ranges are called endemic species. On remote oceanic islands, almost all the native species are endemic. The Hawaiian Islands, for example, have about 1,000 plant species, a small number compared with those at the same latitude in continental Central America. Although all the Hawaiian species, however, are found only there, whereas the species on continents may be much more widespread. Endemic species are much more vulnerable to human activity than are more widely distributed species because it is easier to destroy all the habitat in a small geographic range than in a large one. In addition to diversity among species, the concept of biodiversity includes the genetic diversity within species. One example is our own species, for we differ in a wide variety of characteristics that are partly or wholly genetically determined, including height, weight, skin and eye color, behavioral traits, and resistance to various diseases. Likewise, genetic variety within a plant species may include the differences in individual plants that confer resistance to different diseases. For plants that are domesticated, such as rice, these differences may be of considerable economic importance, for they are the source of new disease-resistant domestic varieties. The idea of biodiversity also encompasses the range of ecological communities that species form. A common approach of quantifying this type of diversity is to record the variety of ecological communities an area may contain. It is generally accepted that an area having, say, both forests and prairies is more diverse than one with forest alone, because each of these assemblages is accepted to house different species. This conclusion, however, is indirect. It is likely based on differences in vegetation, structure, or appearance rather than directly on list of species. Counting species. About 1.9 million species have scientific names. Estimates of the total number of living species cluster around 10 million, which means that most species have not been discovered and described. These estimates omit bacteria because of the practical problems in defining bacterial species. Simply counting species must be at best an incomplete measure of biodiversity, for most species cannot be counted with unreasonable time. At the present rate of describing new species, it will take about 1,000 years to complete the catalog of scientific names. Of the approximately 1.9 million species now described, perhaps two-thirds are unknown from only one location and mainly from examining only one individual or a limited number of individuals. So knowledge of a genetic variation within species is even more constrained from just a few well-studied species. It is clear that genetic variability can be substantial and that it differs in extent between species.
Now, let us talk about measuring diversity through relative abundance. Species diversity is determined not only by the number of species within a biological community, for example, species richness, but also by the relative abundance of individuals in that community. Species abundance is the number of individuals per species, and relative abundance refers to the evenness of distribution of individuals among species in a community. two communities may be equally rich in species, but they differ in relative abundance. For example, each community may contain 5 species and 300 individuals, but in one community, all species are equally common. Example is 60 individuals of each species, while in the second community, one species significantly outnumbers the other 4. So, when measuring diversity, we may use relative species abundance, but how do we define this way? Relative species abundance is a component of biodiversity and is a measure of how common or how rare a species is relative to other species in a defined location or community. Relative abundance is the percent composition of an organism of a particular kind relative to the total number of organisms in the area. So we can calculate it by dividing the number of species from one group by the total number of species from all groups. So why is relative species abundance important? Knowing the abundance of different species can provide insight into how a community functions. Data on species abundances are relatively easy to obtain and may give insight into less visible aspects of a community, such as competition and predation. Using several datasets, Frank W. Preston in the year 1948 argued that species abundances follow a normal distribution, partly as a result of the central limit theorem. This means that the abundance distribution is log normal. Log normal is a distribution that is a continuous probability distribution of a random variable whose logarithm is normally distributed. Now let us talk about the patterns in species abundance. In nearly every community in which species have been identified and counted, distributions are highly skewed, or if one tail is longer than another on a graph. That is, there are many rare species and only a few common species. Although a skewed pattern in species abundance seems to be a universal feature of all communities, it may be more pronounced in some ecosystems. To determine what more and less actually mean, we need to quantify the degree to which such patterns are skewed. One way to deal with skewed abundances is to transform them using logarithms. This is visualized by Preston's log normal distribution of species abundance in the year 1948. He constructed abundance intervals, each interval being twice the preceding one, and graphed the number of species within each interval. So let us refer to this figure, which is the graph that he constructed. When plotted on this log2 scale, patterns in abundance were found to approach a normal distribution. For many communities, a log normal distribution provides an accurate description of the abundances of species in a community, and this distribution is therefore frequently used to model the relative abundance of species in communities. Besides the log normal distribution, there are other mathematical models. Examples are geometric series, log series, and broken stick in use, each incorporating different mathematical assumptions and theoretical concepts of how communities function. Using logarithms to transform data is simply another way of plotting abundance data that facilitates comparisons of the data across communities. However, assuming that abundances in natural communities are indeed log normally distributed has some practical consequences. According to a log normal distribution, not only are there few common species with a high local abundance, but also few species seem to be truly rare. Deviations from a log normal distribution therefore can be used to estimate the completeness of a species inventory and estimate how many more species might be found if sampling is more intensive. As community sampling is always incomplete, this information has significant practical value. Being able to extrapolate information from a limited number of samples improves the effectiveness of species inventories, including monitoring changes in species abundances due to global warming or restoration efforts, for example. So going back to the definition of relative species abundance, it is a measure of number of species in an area. Let us look at these two communities. So, in determining species evenness, we need to count the number of individuals of each species, or how common is each species in the community. Here in our first community, we can see that there are 4 species or 4 types of trees. Species A, B, C, and D. And they are equally even. Each species is 25% of the community. Community 2 also has 4 species, but species A is more common when compared to species B, C, and D. It is 80% of the community. To understand more, we have here the data of species evenness of both communities. All species in Community 1 are equal when it comes to species abundance. Community 1 has high evenness, or most of the species are common. Here is a graph for the Community 2. The bar of species A is much higher compared to the other species, and this represents low evenness, or few of the species are very common and the rest are rare.
Good day everyone, my name is Ami Omega. So today we will tackle about the Shannon Diversity Index. So first off, what is diversity? Diversity incorporates both the number of species in an area and the evenness of their abundances. So later on, I'll show you the step-by-step process of how we will going to have the number of species in an area using the formula of SDI. Okay, so what we have here in this side is the 6 different species that has different community. So the next one is the number of individuals, wherein they have different number of species as well. So for A, we have 49, B, we have 17, C, we have 11, D, we have 10, E, we have 5, and F is 8. So for us to come up to the total number of species in an area, which is the Shannon Diversity Index, we have here is the formula that we are going to use later on. So SDI is equal to 1 minus sigma n parenthesis n take 1 divided by big letter N and then parenthesis n take 1. So the first step, or the step 1 in this process, we're going to have subtract the number of individual with 1. So in this case, we will going to have the 49 as 48, 17 subtract by 1 is 16, 11 is 10, 10 is 9, 5 is 4, and 8 will be 7. So step 2, we're going to have to multiply the number of individual with the number of n take 1 that we have here. So 49 times 48 is total of 2,352. 17 times 16 would be 272. 11 times 10 will be 110. Then 10 times 9 will be 90. 5 times 4 will be 20. And then 8 times 7 will be 56. So next step for that is we're going to total the number of individuals that we have in this specific community. So the total of the individuals will be 100.
So 49 plus 17, 17 plus 11, 11 plus 10, 10 plus 5, and then 5 plus 8 will be the total of 100. And then right here, we're going to have this sigma, wherein it is the summation of the total number of multiplication times and take 1. So the total number in this part will be 2,900. So now we have the enough information for us to have the diversity that we will going to sum later on. Okay, so SDI is 1 minus the total of, or the sigma in this part. So what we have here is 2,900, and then the total number of individual, which is this one, so 100 times n take 1, which is 100 minus 1. So we will going to have 99. And then next step is we're going to divide 1 minus 2,900 divided by the multiplication of 100 times 99 is 9,900. So the answer for this one is 1 minus 0.29, in which when we subtract 1 minus 0.29, we will come up to the answer 0.71. So this simply means that an SDI of these 6 community or 6 different species, there is a 71% chance that an individual select a random would be from different species.